So our presentation is locomotive activity in Drosophila using the dam system. My name is Michelle.
And my name is Tomas. Okay, so the Drosophila activity monitors, also known as the dam system, is used to analyze and record locomotive activity, circadian cycles, and sleep patterns in Drosophilas. Thanks to the dam system, the first gene that controls the daily biological rhythms was discovered, which provides insight into the mechanisms underlying biological clocks across many species. Our project aims to characterize the circadian controlled locomotive activities of a panel of interspecies lines generated from two sister subspecies, which are simulants and chinchilla. Each interspecies line has 12.5% of genetic loci from chinchilla and 87.5% of loci from these simulants. And the reason why we chose these two sisters subspecies is due to the difference in anticipation and difference in overall peak activity. So we collected 3-day locomotive data of about 10 interspecies lines in 5-minute intervals under a 12-hour day and 12-hour night cycle. And the purpose of our research project is to characterize the circadian controlled locomotive activities of these interspecies lines.
Okay, so on the methods section, what we use to basically record the behavior of the flies is as we mentioned, the dam monitor system, which is shown here, a diagram. So 32 male flies were used in each monitor, it looks like this. And each tube had, let me zoom in so you can see better. Okay, so each tube had one fly, and there was an infrared beam light, which recorded how much time the fly went back and forth in the tube. And this was recorded by a PC computer, which was connected to the incubator. So moving on to the results. This table A and B, show the anticipation for the control group simulants and chinchilla and two interspecies which we're comparing to. So as we can see, that simulants has a higher anticipation, and what this means is anticipation means when the fly knows when the light's going to turn on. So they're already having behavior movement before the lights turn on. So simulants had the highest compared to all of them. And as we see, this interspecies with the dark blue line was B112. That one had the lowest anticipation, which also shown on graph B is chinchilla also had low anticipation. So that's basically what it means. And then moving on to this graph right here, this one shows simulants compared to 7 different interspecies. And the reason why chinchilla is not on this one is because when we started our experiments, we found out that our chinchilla was actually not chinchilla, it was simulants all along. So we discarded that from our experiment. So we had only one control group. So these graphs right here that I was talking about before, these were from past students that did the experiment. And so we see that the higher peak shows the activity. So the peak shows how much activity the flies have. And like I was saying, the higher one had more activity than the rest of them. And for example, the species V327G5 had the least activity in a 24 cycle. And then we also did our average anticipation right here, which showed how much anticipation each interspecies had compared to our control group, which was simulants. And graph C is the activity per day in a light to dark ratio. And basically this shows how much activity each fly had. And like we mentioned before, the species V327G5 had the least activity compared to all the other ones, and simulants had the most activity.
Okay, so for our discussions, we established a protocol for characterizing the locomotive profiles of a set of uncharacterized interspecies lines between simulants and chinchilla. Some of these lines had high anticipation and lower locomotive activities, and some of the lines had low anticipation and lower locomotive activity, which basically we concluded that anticipation and locomotive activity does not seem to correlate. And like Michelle said, when we conducted the dam analysis and plotting the data for each interspecies, we concluded that the chinchilla were not actually chinchilla, so they were discarded from our experiment. And our project is in collaboration with the University of Mississippi, so we would like to acknowledge them. We would like to acknowledge Scholars Academy and also Dr. Parker and everybody from our lab, and also Dr. Kang, which was our supervisor.
Oh, wonderful presentation. I do have a question. I want to go to that, the middle group of slides, the last one, the lower one, I should say. So that V327G5, I mean, it did have wings, is that right?
It does, but whenever we were using these interspecies, some of them actually either had shorter wings or and actually they don't really fly in the tubes because the tubes are actually really small. It's more about how much time they pass.
Really?
Red light, yes.
So they're crawling versus flying. Is that what you would say?
Yes, they're crawling versus flying because on one end it has food. We actually have food, and on the other end it has a stopper with
cotton,
so it won't fly away.
Okay, and how long is the tube? Can you tell me that just for a little? I mean, do you have an idea, or did you tell me that already? Like from one side, one length to
the tube is really tiny. It's really short.
We'll make a guess.
I would say like almost your pinky because I don't really know the exact measurements.
Yeah. Okay. So that maybe, I mean, in
length.
inches, right? That'd be like 2 inches.
Yes.
So really very small. Little.
Yeah.
Very, very tiny. And width, I'm just curious, width, how diameter, how?
I'm trying to compare it.
Would it
be a straw?
Okay.
Really, because the fruit flies are really small too.
Yes.
I would say smaller than a straw, like because Michelle said the flies don't fly because they don't have room for them to fly. They, like Dr. Parker said, they only have room for them to move, like you said, crawl.
Okay. Okay, very, very interesting. So give me your anticipated, generally results if you had really had, what's it called? Chinchillian, or how did you say it?
Chinchilla?
Yeah. What would you have anticipated given what you found here? I mean, can you say?
Well, we anticipate.
Yeah. So what we anticipated was simulants having overall higher anticipation and overall higher peak activity versus chinchilla. Chinchilla has very low anticipation and lower overall peak activity. That's why we chose those two sister species to compose our other inbreed interspecies, because due to the difference in their anticipation and their overall peak activity.
And we actually read articles on these. So on the articles, it did say that this was like expected, that the simulants are going to be more active than the chinchilla.
Right. Chinchilla, yeah. So how much work did you do, and what tipped it for you to figure out, no, this is not what we thought we had? This is not the chinchilla.
We had done like 3 rounds already, and the rounds take forever because it's 8 monitors and every monitor has 32 dam tubes. So in total, that's like 256 tubes, and they're so small. So you have to make food for them, put the cotton, put each fly individually. And you have to be careful with the fly because you can't put too much pressure or else they'll like die or you'll squish them to death. So we did 3 rounds, and whenever we saw our data, we saw that the lines, when we compared them in graphs, the lines for simulants and Centralia were like the exact same.
Uh-huh.
Dr. Kang was telling us, I don't think this is just, I'm going to check with, I believe it was Dr. Greg from the University of Mississippi. Like he,
Oh, yeah, because he sent you the flies.
Yeah, and
yes,
also even before we did the data, Dr. Kang told us that the reproductive cycle was going too fast for it to be chinchilla. So she actually predicted it even before we did the actual data and we plotted that.
Wow. I wonder if that, well, it's just an aside. I wonder if that impacted his data. How did he confuse? I was probably a graduate student that confused it. But anyway, okay, y'all, excellent. Keep your recording. It'll go straight to whoever put in the Zoom link, and then I'll ask you for them later. Okay.
okay. Thank you so much,
Very
Dr. Parker.
nice.
Thank you,
Very
Dr.
nice. And let's
Parker.
see, I have maybe 10 more minutes and then you get to go and look at others. Okay.
Okay.
Sounds good.
Thank
you
y'all.
so
Very
much.
good.
Thank you, Dr. Parker.
What is it about?
So, this is our poster on locomotive activity in Drosophila using the DAM system, and DAM stands for Drosophila Activity Monitor. Do you want to do the introduction, Thomas, or do you want me to do it?
Yeah, so basically, the Drosophila Activity Monitor, or the DAM system, is used to analyze and record locomotive activity, circadian cycles, and sleeping patterns in Drosophila flies. And thanks to the DAM system, the first gene that controls the biological rhythms was discovered, which provides insight into the mechanisms underlying biological clocks across many species. And our project aims to characterize the circadian controlled locomotive activities of a panel of interbreed interspecies generated from two Drosophila sister species, Simulans and Chinchilla. And each interbreed line was composed of 12.5% of genetic loci from Chinchilla and 87.5% of loci from Simulans.
So let me zoom in here. This is the model we used to record the behavior of the fly, and this is the DAM system. So it has 32 glass tubes in each monitor, and we use 32 male flies. And basically, whenever we hook this up to in the incubator, there is an infrared light which captures how many times the fly moves back and forth. That is the activity. And this is recorded by the computer that it is connected to. And the most important thing about our experiment was the activity anticipation that each fly had. So graph A and graph B are the anticipation graphs for the fly. So we see that it compares Simulans and Chinchilla versus the two interspecies line. And we see that Simulans has, which is the black line, has a higher anticipation and peak. And basically what this means is whenever a fly has a higher anticipation or low anticipation, that means that it anticipates when the light's going to be on. So way before the light's going to be on or off, it already starts to have movement in the tube. And when it has low anticipation, which is the other lines, the light blue and dark blue, it means that it doesn't actually move at all before the lights turn on. It actually does it as soon as the lights turn on. That's why we see the peak go high. And the peak shows how much activity they have. And this is recorded over 24 hours. And then this graph right here with 7 lines is what we recorded. This the dark blue line shows Simulans, which we can see has a higher anticipation and higher activity based on the peak. And compared to all of them, also V327G5 is an example of a fly that has not much activity nor not much anticipation. And we also did our bar graph comparing the anticipation with all these 7 interspecies lines here. And Graph C shows the activity per day in light to dark ratios. And basically this graph shows how much activity they have in a 24-hour period as well. So this is one day. And we see that simulants has the higher activity, locomotive activity compared to all the other ones. So like I said earlier, V327G5 has the least activity counts per day.
And so basically for our project, we established a protocol for characterizing the locomotive profiles of a set of uncharacterized interspecies lines between the simulant and chinchillas. And when conducting the dam analysis and plotting the data, we concluded that the chinchilla flies that we were using in our experiment were not indeed chinchilla, so they were discarded from our experiment. And also, some lines have high anticipation but low locomotive activity, and vice versa. So we concluded that the locomotive activity and anticipation do not seem to correlate.
And these are our acknowledgements. Do you have any questions?
No. Cool project. That's really interesting.
Thank you so much for listening.
Thank you.
Yeah, thank you for presenting. Have a good day.
You too.